Welcome back guys. Our next topic is hereditary spherocytosis. It is an autosomal dominant disorder, as the name suggests, hereditary. So it will affect all the progeny of an affected family. All sons and daughters of same parents will get this disease, which occurs due to intrinsic defect in the cell membrane of RBCs, which results in extravascular hemolysis. So RBC will be destroyed outside the blood vessels, and that site is most commonly spleen, which will result in normocytic anemia with high reticulocyte count. So here is the concept. Red blood cells contain cell membrane like the normal cells. The cell membrane of RBC contain a specialized protein which is known as spectrin. It has two chains, alpha and beta chain. Both of these chains are attached to another protein which is known as ankyrin, which is attached to another protein anchored in the cell membrane which is known as band 3. On the other side, this spectrin molecule is attached to a protein known as actin, which is further attached to another anchored glycoprotein which is known as glycophorin A. And this spectrum of these proteins are repeated in the same manner on the surface of RBC, which results in a meshwork of a structure that maintains the biconcave shape of these RBCs. But when there is mutation in any of these proteins, especially ankyrin protein, it will result in defective structure of this meshwork, which will cause the RBCs to lose its biconcave shape and form a round cell which we call spherocyte. As it is occurring due to a genetic mutation, we call it hereditary spherocytosis. These RBCs are called spherocytes because they are spherical in shape, hyperchromic red cells, and it lacks the central zone of pallor. And due to the structure defect in the membrane of these spherocytes, the normal sodium-potassium pump activity is lost, which causes the loss of potassium and water from these cells. As a result, the size of these RBCs becomes shrunken and the size and the volume of these RBCs become less. But the normal amount of hemoglobin is same, so the mean corpuscular hemoglobin concentration (MCHC) is raised. So what's the problem with these RBCs? Actually, they may function the same or slightly less than the normal RBC. What's the real problem is that when these spherocytes pass through the vasculatures of spleen, they cannot make their way through that and their membrane ruptures, causing the extravascular hemolysis of RBCs, as this hemolysis is not occurring inside the vessels but inside the spleen. So it is extravascular hemolysis, and due to accumulation of these phagocytes and the iron releasing from these rupturing spherocytes causes the increase in size of liver and spleen, resulting in hepatosplenomegaly, and the lifespan of normal RBCs is reduced from 120 days to mere 10 to 20 days. So the patient shows anemia. Also, in response to that, the bone marrow tries to form increased number of RBCs, and in response to that, it releases a large number of reticulocytes into the blood circulation, which causes reticulocytosis in peripheral blood. So here is the histological illustration of hereditary spherocytosis. As you can see, these are the normal RBCs and these are the reticulocytes, and these small but hyperchromic cells are spherocytes. And look carefully, these cells lack the central pallor. Instead, these are hyperchromic. And this is a real histological slide of hereditary spherocytosis. These are normal RBCs, these are biconcave cells, and these are reticulocytes, and these are spherocytes. At this point of this video, I hope you have gained a clearer understanding of the concepts we have covered. For those of you who want to reinforce your learning and dive deeper into this topic, we have created comprehensive review notes. These notes break down the key points discussed in this lecture, making it easier for you to grasp the material quickly. Be sure to check them out by clicking the link in the description below. Happy studying. And here is a summary of all these events. Microscopic features shows spherocytes. Lifespan of RBCs is reduced to mere 10 to 20 days. Reticulocytosis is prominent. There is marrow erythroid hyperplasia because bone marrow tries to make more RBCs, and there is deposition of iron, hemosiderosis in liver, spleen, and bone marrow. The gross features are patient may or may not show pallor. Jaundice is present because of increased turnover of these RBCs, and lastly, hepatosplenomegaly. Let's conclude the session with this diagnostic test. We used osmotic fragility test to test these RBCs. Very important. Normally, the RBC can withstand a range of hypoosmolar solution. But when a spherocyte is put in that hypoosmolar solution, it bursts into pieces because the cell membrane of these RBCs is very fragile. So the osmotic fragility test results in the bursting of these spherocytes. And the second most important diagnostic test is when you do the CBC of these patients. These show increased MCHC value. And whenever you see increased MCHC value in CBC, the hereditary spherocytosis should be in the top of list in your mind. Again, very important. And this concludes our session of hereditary spherocytosis. Thank you.